Hello everyone, we are group 2124 and our team has worked with BioRacist for the past 2 semesters. This is actually the 4th year that BioRacist has worked with senior design teams for their implantable glucose sensor. In 2017, the senior design team was tasked to create a smartwatch to read the signals from their implantable sensor. In 2018, the team was tasked to improve upon the design from the previous team. And in 2019, the group was tasked to create cleaner and more usable data points. The problem BioRacist is facing is the test bench they were using produced large amounts of electrical noise, rendering any lab experiment with an implantable device useless. So, this in mind, our job that was given by BioRacist was to solve this problem by making a new test bench that was able to mix 2 different glucose solution contents together in a controlled, measurable fashion.
Hardest problems we had to work around this year was COVID-19. These restrictions limited our in-person work with one another, delaying the start of the physical work on our project together. In semester 1, we worked on RF shielding the equipment inside of the BioRacist lab. This included shielding data cables, stepper motor casing, and most important and impactful, shielding the ring stand in which the implantable sensor is mounted to. This drastically reduced the noise affecting the device, making the data from the experiment once usable for the first time. However, we continued to work towards our original goals. We began semester 2 with researching data sheets and looking into torque specifications on stepper motors along with low noise output stepper motor drivers. After further research, we as a group decided between 2 possible motors and 1 driver. The 2 motors we chose were the NEMA 8 and the NEMA 17 stepper motor. After operational testing of the NEMA 8, we found the motor was exceeding the operational temperature limit of 130 degrees Fahrenheit when the motor was powered at max current while operating the peristaltic pump. This moved our current and final motor choice of the NEMA 17 stepper motor, a larger and higher current rated motor. We continued to stick with our original choice of the stepper driver, which is the DM320T low noise driver.
So for our mechanical challenges, once we finished selecting a motor and motor driver, we began working the test bench layout and the coupling of our 2 motors to each of the pumps. The previous test bench relied on a rod pressed into a spring to couple the motor to the pump, and we decided to move away from that design, and we filed down the motor shaft into a rectangle that fit snugly into the input of the peristaltic pump. And during the final assembly of the test bench, we found out that our motor mounting bracket wasn't adjustable, and we had to manually align the center of the filed motor shaft with the center of the pump. And because we were now using a direct connection of the 2 shafts, we solved the problem by adding a 1/2-inch wind spacer to the bottom of our bracket. For programming challenges, apart from learning how to tell our Arduino to talk to the motor drivers, there were 2 major areas that we had to focus on. And the first was maintaining a 1 milliliter per minute flow rate between the 2 pumps at the same time, meaning as one pump increased in flow rate, we had to equivalently decrease the other pump to maintain that 1 milliliter per minute. And the second area was creating a well-documented program that allows our parent company BioRacist to adjust certain variables to run the program for different periods of time, and also decrease and increase the 2 different pumps for different time parameters.